everyone, Namaskar and welcome back to my channel Learn with Sis. I'm really glad that you could join me today as we take a closer look at the important topic of protein. In today's session, we will explore everything you need to know about the protein, its role in our body and how it keeps us healthy. So, keep watching my video until the end. Here is an overview of what we are covering today. So, introduction to protein and its key sources, the different classification of proteins, the essential function of the protein source in our body, how protein is digested, absorbed and metabolized, and the last one is common disorder and disease related to protein deficiency. So, let's start. Protein is a macronutrient and are large molecules made up of amino acids. It is essential for building and repairing tissues, supporting immune function and producing enzymes and hormones. Protein are found in every cell of our body. Like carbohydrate and fats or lipids, it contains carbon, hydrogen, oxygen, but they are unique in that they contain nitrogen. The normal protein requirement for an adult is 1 gram per kg of body weight. However, growing children's disease condition as well as pregnant and lactating mothers requires more protein. The normal range of serum protein is 60 to 85 gram per liter or 6 to 8.5 gram per deciliter. So let's move to the classification of the protein. So protein are classified in different ways, but I have listed some classification here. So number one, I have listed classification based on sources or origin of protein molecules. So that is animal proteins and plant protein. So let's move to the animal protein. These are the protein derived from animal sources such as egg, milk, meat and fish. They are usually called higher quality protein or class 1 protein because they contain adequate amount of essential amino acid. Whereas plant protein are called lower quality proteins or class 2 protein because some essential amino acid is missing. The 4 common limiting amino acids are methionine, lysine, threonine and tryptophan. So now move to another classification that is based on shape of the protein molecules. On the basis of the shape of protein molecules, protein have been grouped under 2 categories, that is globular protein and fibrous protein. Globular proteins are more spherical or globe-like structures. These structures involve them to be more soluble in water. Examples are hemoglobin, myoglobin, immunoglobulin, amylase, insulin and HGC. Now fibrous protein. These proteins have elongated fibrous or sheath-like structures such as keratin, collagen, elastin and fibrin. They mainly serve structural or protective layer of the cell and it is insoluble in water. So now move to the classification based on composition and solubility. So in composition, protein can be classified in 3 ways. One is simple protein, conjugated protein and derived protein. So simple protein is made up of amino acid linked by peptide bond. They yield only amino acid when broken down. Example includes albumin, globulin and euglobulin. These are water soluble, whereas protamine is insoluble in water. So now move to next, conjugated protein. It contains amino acid with additional non-protein substance such as lipid, vitamin, carbohydrate and metal, etc. Example of conjugated proteins are lipoprotein, glycoprotein, phosphoprotein, hemoprotein, flavoprotein, metalloprotein, etc. Another one is derived protein. These are the protein which is derived from simple or conjugated protein through the physical or chemical process. Derived protein is further divided into two types, that is primary derived protein and secondary derived protein. Primary one is insoluble in water. Example are denatured protein, metaprotein and conjugated protein. Whereas secondary derived proteins are soluble in water. Examples are proteases, peptides and polypeptides. Let's move to the classification based on biological function. Protein has a multiple biological function, including acting as an enzyme and hormones, maintaining proper fluids and acid base balance, providing nutrient transport, making antibodies, enabling wound healing and tissue regeneration, and also providing energy when carbohydrate and lipids intake is inadequate. You can see the table here. Classification based on biological function. Now, another one is classification based on amino acids. We know that amino acid is a building block of protein. N is organic compounds with amine group minus NH2 and a carboxyl group minus COOH in a unique side chain. They contain carbon, hydrogen, oxygen, and nitrogen. These are classified into three groups, which are essential amino acid, non-essential amino acid, and conditional amino acid. Normally, there are two types of amino acid. One is essential and non-essential, but conditional amino acid are usually non-essential amino acid, but become essential under certain condition like illness or stress. And we know that essential mean body cannot synthesize, but it must be obtained from the diet. And non-essential can be synthesized by our body. You can see the table here. Now again, move to another classification that is based on structure. There are four types based on structures. They are primary structure, secondary structure, tertiary structure, and quaternary structure. The primary structure of the protein refer to the linear sequence of amino acids in the polypeptide chain. The example of primary structure of the protein is the first six amino acid in hemoglobin. That is valine, histidine, leucine, threonine, proline, and glutamate. Whereas secondary structure is a repetitive 3D structure of the protein. The most common type of secondary structure are the helix and depleted sheet. Coming to the tertiary structures, the tertiary structure of the protein refer to the overall three-dimensional arrangement of its polypeptide chain in space. Example of protein with tertiary structures include enzymes, antibodies, and hemoglobin. Now, last one is quaternary structures. The quaternary structure of the protein is association of several protein chains or subunits into a closely packed arrangement. Example of the proteins with quaternary structures include hemoglobin, DNA polymerase, ribosomes, antibodies, and ion channels. Let's move to the function of protein. Protein has lots of function. Whenever the body is growing, repairing or replacing tissue, protein are involved. So I'm going to list out some important function here. It is similar like a biological functions. Protein is an important source of energy. Each gram of protein provides 4 kilocalorie. So it is energy giving. Another one is it work as a structural material. It work as an enzyme. It work as a hormone. It is important for acid base balance. So the most important function is it helps in maintenance and growth. It works as a transporter or carrier proteins. It works as a defense protein. It works as a nutrient and storage protein. Another one is contractile or motile protein. And another one is toxic protein. So you can see the function here. Let's move to the digestion, absorption, and metabolism of protein. Protein digestion refers to the process by which larger molecules of the protein, that is polypeptides, is broken down into the simpler one, that is amino acid. So there are many factors that affect protein digestion. Some key factors are protein sources. Animal protein are generally more easily digested than plant products. Cooking protein can make them easier to digest. Enzyme activities, enzyme like pepsin and trypsin can influence how well protein are broken down. Next one is digestive health. Condition like low stomach acid or digestive disorder can impair the protein digestion. Meal composition. The presence of fat, fiber, or carbohydrate can either slow down or enhance protein digestion. Age. Increasing age can decrease the efficiency of digestive processes. So it makes harder to digest protein. So let's move to the digestion of the protein. After ingestion of protein containing food, that is polypeptides, food directly move to the stomachs through esophagus by peristalsis movement is because there is no protein splitting enzyme secreted in the mouth. Only mechanical breakdown of the proteins occur at mouth. So the large polypeptides of the proteins reach to the stomach. Pepsin is present in the stomach in inactive form. Once the protein food is reached to the stomach, it get active with the help of HCl. So you can see the reaction here. Renin, an important gastric proteases, is only produced during infancy and childhood. Renin with calcium helps to thicken or coagulate milk protein casein. This slows the movement of milk nutrient from the stomach, allowing additional digestion time. So the partially breakdown of protein or peptides then pass into the small intestine where the peptides bond are hydrolyzed by the variety of enzyme. We know that duodenum, the part of small intestine, receive bile from gallbladder and pancreatic enzyme from pancreas. So you can see the pancreatic enzyme here. Pancreatic enzymes are trypsin, chymotrypsin, elastase, carboxypeptidase. So these are in inactive form. You can see on board. And we have intestinal aminopeptidase, dipeptidase, tripeptidase. Bile salt help to make the favorable environment by neutralizing the food with the help of intestinal enzyme and pancreatic enzymes. Polypeptides or smaller peptides are broken down into simpler form, that is amino acid. Beside amino acids, there are other products which are ready for absorption. They are larger peptides, tripeptides, and minute amount of whole protein. Now move to protein absorption. Protein absorption refers to the process by which the body takes in amino acid from the digested protein into the blood stream after they have been broken down into the digestive system. There are certain factors that affect protein absorption. They are protein quality. Complete proteins like animal proteins are absorbed better than incomplete protein, plant-based one. Digestive enzyme activities. Efficient enzyme like pepsin and trypsin are essential for breaking down protein. Health condition also, digestive disorder and low stomach acid can impair absorption. And cooking method as well. Proper cooking helps to enhance Absorption, hydration. Proper hydration support digestion and absorption. Other nutrient like deficiency in nutrient like B6 or magnesium can hinder protein absorption. So let's see how smaller protein move into the blood.
You can see here different form of digested food ready for absorption.
Free amino acids enter into the epithelial cell by secondary active transport coupled to sodium, that is sodium dependent active transport. This transporter required sodium ions to facilitate the active transport of amino acid into enterocyte. Short chain of 2 or 3 amino acid, that is di or tripeptides, are absorbed by a secondary transport that is coupled to hydrogen ion gradients. It means that it enters through the cross border of the intestinal mucosal cell, where they are hydrolyzed to free amino acid with the help of cytoplasmic peptidases. These are transported into the bloodstream through a facilitated diffusion carrier in the basolateral membrane. Relatively larger peptides may be absorbed intact either by uptake into the mucosal epithelial cell, that is transcellular route, and by passing in between the cell, that is paracellular. Minute amount of whole protein enter into the epithelial cell through pinocytosis method and leave the cell through exocytosis process. So this is how the digested product of protein enter into the blood. You can see here. Now let's move to the protein metabolism. Protein metabolism is the process by which the body breaks down dietary proteins into the amino acid, uses them to build new protein for growth, repair, and function, and eliminate waste products. So, there are certain factors that affect metabolism. They are diet, exercise, hormones, age, health condition, hydration, nutrient deficiency, sleep, and stress. The metabolism of protein complete by two processes, anabolism and catabolism. Liver is the central processing unit for protein metabolism. The protein synthesis is accomplished through transamination and deamination reaction. Transamination is the protein synthesis reaction. So, transamination is the protein synthesis reaction. So, in transamination, the nitrogen from one amino acid is used to manufacture another amino acid and form a long chain of amino acid and store as a protein in a liver. In deamination, the amino group is removed from amino acids and converted to ammonia in the kidney. The ammonia created during deamination is toxic to the body. So, enzyme in the liver convert ammonia into urea. Approximately 100 gram of dietary protein is consumed to produce about 17 gram of nitrogen after its metabolism. Out of 17 gram of nitrogen, 16 gram is eliminated from urea through the urine. Therefore, the greater the extra protein consumed, the greater the production of ammonia, that is urea. The remaining carbon structure in the deamination process is either used for energy or transport to the liver for gluconeogenesis and stored as a fat or converted to the glucose. So this is all about the protein metabolism. Now let's move to the end part of our video, that is protein deficiency. Deficiency of energy or protein commonly occur in developing countries like Nepal, Bangladesh, and India. This is manifested as a marasmus and kwashiorkor. Protein energy malnutrition is the term used to describe clinical disorder resulting from a varying degree of energy and protein deficiency. In our next video, I will bring more information about it. So another deficiency is protein C and S deficiency. Both are congenital or acquired condition that lead to increased risks of thrombosis. Research indicate that 1 in every 300 people suffer from the lack of protein C. Similarly, protein S deficiency affect 1 in 20,000 people. So this is all about protein. We discuss in different topic. Hope you like my videos. Thank you for watching my video. So don't forget to like, share and subscribe. Thank you.